Hi, today we are discussing about the syllabus covered. So first topic is new drug development and approval process. The contents of the drug discovery are: first, drug discovery and drug design; second, biological characterization; third, early formulation studies; fourth, the investigational new drug application, that is IND; and the last is the new drug application, that is NDA. The Federal Food, Drug and Cosmetic Act, as regulated through Title 21 of the US Code of Federal Regulation, requires a new drug to be approved by the Food and Drug Administration before it may be legally introduced into interstate commerce. To gain approval for marketing, a drug sponsor, for example a pharmaceutical company, must demonstrate through supporting scientific evidence that the new drug or drug product is safe and effective for its purpose use. The process and time course from drug discovery to approval for marketing can be divided into following process. Firstly, new chemical entity, then pre-formulation studies, then formulation studies, investigational new drug application, preclinical and clinical studies. In that, phase 1, phase 2, phase 3 and new drug application. The drug discovery and drug design. First, the source of new drug, a goal drug, method of drug discovery, a lead compound, and prodrug. The next chapter is phases of clinical trial. Introduction. A clinical trial is a systematic investigation in human subjects for evaluating the safety and efficacy of any new drug. The clinical trial are a set of tests in medical research and drug development that generate safety and efficacy data for health interventions in human beings. Clinical trials are conducted only when satisfactory information has been gathered on the quality of the non-clinical safety. Health Authority Ethics Committee approval is granted in the country where approval of the drug is sought. Clinical trial is a mainstay for bringing out new drugs to the market. The next point is drug review steps. There are mainly 8 steps in the drug review that are as follows. First is preclinical testing. Second, an investigational new drug application. Third is phase 1 studies. Fourth is phase 2 studies. Fifth is phase 3 studies. Sixth is submission of new drug application, is the formal step asking the FDA to consider a drug for marketing approval. Next is FDA reviewer will approve the application or find it's either approvable or not approvable. Then last is phase 4 studies. The drug review. Before one can initiate testing in human beings, extensive preclinical or laboratory research is required. Research usually involves years of experience in the animal and human cells. The first is preclinical evaluation phase, that is animal studies. The major areas in the preclinical evaluation is the pharmacodynamic studies, in vivo in animals, in vitro in preparation. Next is absorption, distribution, elimination studies, then acute, subacute, chronic toxicity studies, and the therapeutic index. The filing of IND application are: once the preclinical studies have indicated the safety and efficacy of a drug, an IND application has to be filed with the regulatory authorities. The contents of IND application. First is clinical data, then information on composition and source of drug, then chemical and manufacturing information, then proposed clinical plans and protocols, ethical committee clearance. Then the phases. First is phase 0 studies, that is microdosing. Study of new drug in microdoses to derive pharmacokinetic information in human before undertaking phase 1 studies is called as a phase 0. Microdose means the less than 1 divided by 100 of the dose of a test substance calculated to produce pharmacological effect with a maximum dose of less than 100 micrograms. Objectives are to obtain preliminary pharmacokinetic data. The advantages of phase 0 is less chances of adverse effect, short duration, less number of volunteers, reduced cost of development, and reduced drug development time. and the limitations are study mainly based on pharmacokinetic parameters, not efficacy and safety based. Then next is phase 1. Phase 1 is the first stage of testing in human being subjects. The design is designed to assess the safety, tolerability, pharmacokinetic and pharmacodynamic of the drug. It contains the 20 to 25 healthy volunteers. The duration of this phase is 6 to 12 months. There are mainly three types of phase 1. First is SAD, that is single ascending dose studies. The next is MAD, that is multiple ascending dose studies. Then food effect, the investigates differences in absorption caused by the food. Then next is phase 2. It is a therapeutic exploratory trial, consists of 20 to 300 subjects. This phase contains to confirm the effectiveness, monitor side effects and further evaluate safety. First in patients, the duration of this phase 2 is 6 months to several years. There are two types of phase 2. Phase 2A, that is designed to assess dosing requirements, and phase 2B, that is for design to study efficacy. The next phase is phase 3. It is a therapeutic confirmatory trial. The target population of is 100 to 3000 patients. Duration of this phase is it take a long time, up to 5 years. Then next is phase 4, that is post marketing surveillance phase. It has a no fixed duration and no patient population. It helps to detect the rare ADRs, drug interactions, and also to explore new uses for drugs, sometimes called as a phase 5. The clinical trials is a human experiment designed to study the efficacy and safety of a new drug interventions. Next chapter is Introduction and History of the Pharmacovigilance. The pharmacovigilance WHO definition is the science and activities relating to the detection, assessment, understanding and prevention of adverse effect or any other drug related problems. The aim and scope of pharmacovigilance. First is patient care. To improve the patient care and safety in relation to the medicines and all medical and paramedical interventions. Then public health. To improve public health and safety in relation to the use of medicines. Then risk benefit assessment, to contribute to the assessment of benefit harm, effectiveness and risk of the medicines. And the last is communication, to promote the understanding, clinical training and effective communication to health professional and the public. The objectives of the pharmacovigilance. The objective of pharmacovigilance is the reduction of the frequency and the severity of the adverse effect of a drug while maintaining or better improving their efficacy. The quality of the drugs more particularly depends on the pharmaceutical industry and the authorities responsible for their approval on the market and their withdrawal if necessary and the recommendation of their use. Then next point is who will report the adverse event. Adverse event can be reported by the physician, hospitals, pharmaceutical manufacturers through the medical representatives and pharmacist or nurse. Then the next chapter is regulatory authorities and different forms of reporting in different countries. The reporting from the India is done by the suspected adverse drug reaction and reporting form. Then the yellow card scheme is used in the UK system for collecting the information on suspected adverse drug reaction to the medicines. In USA, the MedWatch form is filled for the adverse drug reaction. Then the divisions of pharmacovigilance. First is evaluate, then monitor, analyze, action and communicate. These are the divisions of the pharmacovigilance. Then the softwares used for the reporting of ICSR to health authorities. In USA, the PharmaReady is the software used for the reporting. In Europe, it's EVWeb. In India, the Sugam. In MHRA, the Vigilance. And for all raw regions, our manual submissions are done. Next chapter is Guidelines on Good Pharmacovigilance Practices. There are mainly 14 modules are available in the pharmacovigilance system. The first module is pharmacovigilance system and their quality system. Second module is pharmacovigilance system master file. The third module is pharmacovigilance inspection. Fourth module is pharmacovigilance audits. The fifth module is risk management system. Then the sixth module is collection, management and submission of the reports of suspected adverse reaction to medicinal product. Then seventh module is periodic safety update report. The eighth module is post authorization safety studies. Ninth module is signal management. 10th module is additional monitoring. The 11th module is safety communication. And the 12th module is risk minimizing measures, selection of tools and effectiveness indicators. So these are the GVP modules. So these are the guidelines on the good pharmacovigilance practices by the European Medical Agency. Next chapter is ICSR case processing, that is individual case safety reports. There are mainly three types of cases. First is valid cases, that is the case report will be 4 minimum valid criteria. First is identifiable patient, then identifiable reporter, suspect drug and adverse event. The next type of case is invalid cases. If out of 4 criteria, if one of the criteria is missing, then case is invalid. The incomplete case means the source document is only having the suspect drug and adverse event. The report types are the spontaneous report, literature and study. The next chapter is adverse event reporting. The adverse event means it's any unfavorable and unintended sign, symptom or disease which are temporarily associated with the use of medicinal product, whether or not considered relating to the medicinal product. Adverse drug reaction. All nauseous and unintended responses to a medicinal product related to any dose should be considered adverse drug reaction. A response to a drug which is nauseous and unintended and which occurs at dose normally used in men for prophylaxis, diagnosis or therapy of disease or for medication of physiological function is called as a regarding marketed medicinal product. Then unexpected adverse drug reaction. An adverse reaction, the nature or severity of which not consistent with the applicable product information. Next point is seriousness criteria. The serious adverse event definition is an serious adverse event or SAR is any untoward medicinal occurrence that result in death, is life-threatening, requires inpatient hospitalization or prolongation of existing hospitalization, result in persistent or significant disability, incapacity, is a congenital anomaly, birth defect, or is a medicinal importance. Next, the steps of ADR monitoring. Steps involved in ADR monitoring are identifying adverse drug reaction, assessing causality between drug and suspected reaction by using various algorithms, then documentation of ADR in patient medical records, reporting serious ADRs to pharmacovigilance centers, ADR regulating authorities. The outcomes of the adverse event. First is fatal, then continuing, recovering, recovered, and unknown. Next, the significance of ADR monitoring. To enhance the patient safety, to keep record of efficacy. Significance of ADR monitoring content: the provide optimal information to users, identifying new information about hazards associated with the medicines, then evaluate the changes in benefit and risk, monitor impact of action taken, and last one is identify previously unknown hazards. There are mainly two types of ADR reporting. First is expedited, that is quick reporting, and routine reporting. Next chapter is action taken with the D-challenge and re-challenge. Action taken, it is the action taken with the drug with respect to the event. For example, patient is taking omeprazole 20 mg, increasing it 40 mg and develop the hyponatremia, result in its withdrawal. Action taken is drug discontinued. The different action taken are drug withdrawn, discontinued, drug increase, dose decrease, treatment period completed, temporarily withdrawn, dose previously discontinued, no change, unknown, and not applicable. The next point is D challenge. This is an evaluation done to assess the ADR when the drug is stopped or discontinued. The term abated or positive D challenge is used when we assess that the ADR adverse event is resolved when the suspect drug is stopped. The scenario of the D challenge is if yes, then yes indicates a complete or partial disappearance of the ADR when the drug is withdrawn or dose is reduced, regardless of the reason for discontinuation or the dose reduction. If no indicates, then its continued presence of the ADR after withdrawn or dose reduction. It is also known as a negative D challenge. And unknown means when information is not available. The D challenge is not applicable if patient received the treatment of ADR, fatal outcome, drug discontinued prior to the event onset, if action taken with the drug is not changed or dose increase, drug exposure during pregnancy, medication error and overdose. Then the next point is rechallenge. Rechallenge, this is an evaluation dose to assess the ADR when the drug is restarted after a positive D challenge. Rechallenge is only applicable when we observe the positive D challenge. The scenario of the rechallenge are if yes, then the ADR repaired upon reintroduction to the suspect drug. It is also called as a positive rechallenge. If no, then the ADR will not appear upon reintroduction of the suspect drug, that is negative D challenge. And if unknown or not applicable, if D challenge is unknown or not, by default or rechallenge is not applicable. The rechallenge is not applicable when the drug exposure during pregnancy, medication error and overdose. Then the action taken with the product and corresponding results. If action taken is a withdrawn with the product, then the D challenge result is positive, negative and unknown, and rechallenge result is positive, negative and unknown. If action taken with the product is unknown, then the both D challenge and rechallenge are unknown, not applicable. If action taken with the product, the treatment period completed, then the D challenge results and rechallenge results are the same, that is not applicable. The action taken with the product is temporarily withdrawn, then D challenge is positive, negative, unknown, and also rechallenge is also positive, negative and unknown. If action taken with the product is not applicable, then both D challenge and rechallenge are not applicable. Same, if no change action taken with the product, then the D challenge and rechallenge also not applicable. Action taken with the product is dose previously discontinued, then D challenge result and rechallenge result are not applicable. Next, the action taken with the product is dose increase, then the D challenge result is not applicable and rechallenge result is also not applicable. If dose decrease, then the positive, negative and unknown, and the rechallenge result is also positive, negative and unknown. Thank you so much.